Bismillahirrahmanirrahim. In this lesson, I will describe about passage of molecules into and out of cell. So diffusion is the movement of molecules from an area of high concentration to the area of lower concentration, which means a long concentration gradient. The molecules of any substance, solid, liquid or gas, are in motion when that substance is above 0 degrees Kelvin or minus 273 degrees centigrade. In a substance, majority of the molecules move from higher to lower concentrations. Although there are some that can move from low to high. The overall or net movement is thus from high to low concentration. Eventually, the molecules reach a state of equilibrium where they are distributed equally throughout the area. So diffusion is one principal method of movement of substance from cells and within cells. Carbon dioxide, oxygen, glucose can cross cell membranes by diffusion. Gas exchange in gills and lungs occurs by this process. Movement of glucose molecule from small intestine into blood capillaries of villi is another example of diffusion. Now I will describe about passive transport. Because a cell does not expend energy when molecules diffuse across its membrane, the diffusion is type of passive transport. The next one is facilitated diffusion. Many molecules do not diffuse freely across cell membranes because of their size or charge. Such molecules are taken into or out of the cell with the help of transport protein that is present in cell membrane. When a transport protein moves a substance from higher to lower concentration, this process is facilitated diffusion. The rate of facilitated diffusion is higher than simple diffusion. Facilitated diffusion is also a type of passive transport because there is no expenditure of energy in this process. The next one is osmosis, which is the movement of water across a semi-permeable membrane from a solute of lesser solute concentration to a solution of high solute concentration. The rules of osmosis can be best understood through the concept of tonicity of a solution. The term tonicity refers to the relative concentration of solutes in the solution being compared. A hypertonic solution has relatively more solute. An isotonic solution have equal concentration of solutes, and a hypotonic solution has relatively less solute. When an animal cell such as red blood cell is placed in an isotonic solution, the cell volume remains constant because the rate at which water is entering the cell is equal to the rate at which it is moving out. When a cell is placed in a hypotonic solution, water enters and cell swells and may rupture like an overfilled balloon. Similarly, an animal cell placed in a hypertonic environment will lose water and will shrink in size. So, in hypotonic environments, freshwater animal cells must have ways to prevent excessive entry of water, and in hypertonic environments, seawater, for example, seawater, they must have ways to prevent excessive loss of water. Water balance problems are somewhat different from plant cells because of their rigid cell walls. Most plant cells live in hypotonic environment. That is, there is low concentration of solutes in extracellular fluids than in cells, As a result, water tends to move first inside cell and then inside vacuole. When vacuole increase in size, then cytoplasm presses firmly against the interior of cell wall, which expands a little. Due to strong cell wall, plant cell does not rupture but instead becomes rigid. In this condition, the outward pressure on the cell wall exerted by internal water is known as turgor pressure, and the phenomena is known as turgor. In a hypotonic solution, the plant cell will be turgid, while in isotonic solution, the plant cell will be flaccid. Because in isotonic environment, the net uptake of water is not enough to make the cell turgid and it will be flaccid, it will be loose. And in hypotonic environment, a plant cell loses water and cytoplasm shrinks. The shrinking of cytoplasm is plasmolysis. Hence, this cell will be plasmolysed. Osmosis and guard cells. Stomata openings in leaf epidermis are surrounded by guard cells. During daytime, guard cells are making glucose and so are hypertonic, having higher concentration of glucose than their nearby epidermis cells. Water enters them from other cells and they swell. In this form, they assume a rigid board shape and a pore is created between them. At night, when there is low solute concentration in guard cells, water leaves them and they become flaccid. In this form, both guard cells rest against one another and the opening is closed. The turgor of cells is responsible for maintaining shapes of non-woody plants and soft portions of trees and shrubs. Application of knowledge about semi-permeable membranes. The knowledge about semi-permeable membranes is applied for various purposes. We know that the semi-permeable membrane is capable of separating substances. Artificially synthesized semi-permeable membranes are used for the separation of bacteria from the viruses. Because viruses are too small and bacteria cannot cross a semi-permeable membrane. In advanced water treatment technologies, membrane-based water filtration systems are used. In this process, semi-permeable membranes separate salts from water, that is reverse osmosis. Osmosis. Filtration is a process by which small molecules are forced to move across semi-permeable membrane with the aid of hydrostatic water pressure or blood pressure. For example, in the body of an animal, blood pressure forces water in dissolved molecules to move through semi-permeable membranes of the capillary wall cells. In filtration, the pressure cannot force large molecules such as proteins to pass through the membrane pores. Active transport is the movement of molecules from an area of lower concentration to the area of higher concentration. This movement against the concentration gradient requires energy in the form of ATP. In this process, carrier proteins of cell membrane use energy to move the molecules against the concentration gradient. For example, the membranes of nerve cells have carrier proteins in the form of sodium-potassium pump. In a resting or not conducting nerve impulse nerve cell, this pump spends energy ATP to maintain higher concentrations of potassium ions and lower concentration of sodium ions inside the cell. For this purpose, the pump actively moves sodium ions to the outside of the cell where they are already in a higher concentration. Similarly, this pump moves potassium ions from outside to inside the cell where they are in higher concentration. The next one is endocytosis. It is the process of cellular ingestion of bulky material by infoldings of cell membrane. So here is cellular ingestion and here is bulky material. The two forms of endocytosis are phagocytosis and pinocytosis. Phagocytosis is cellular eating and pinocytosis is cellular drinking. In phagocytosis, cell takes in solid material while in pinocytosis, cell takes in liquid in the form of droplets. The last one is exocytosis. In this process through which bulky material is exported out, and this process adds new membrane which replaces the part of cell membrane lost during endocytosis. And now it's time to discuss the home tasks for the day 1. These are the home tasks. You have to read and memorize concepts about prokaryotic cell as open system, diffusion, facilitated diffusion, osmosis, tonicity of animal and plant cell filtration, active transport, and exocytosis from textbook pages 66 to 74. And you have to draw the diagrams. Thank you and have a nice day.